herbatę i to nie, ale powiedzmy, że zasadniczo to jest ciągłe to moje mówienie. W jakiej formie wolicie, żeby mówić? Znaczy ja bez przerwy nie dam mówić 1,5 godziny, bo głos mi wysiadł, ale chodzi mi o to, czy wolelibyście, żeby na przykład mówiła pół godziny, robiła przerwę i jeszcze 2 krótkie przerwy, czy tak jak teraz mówię, że powiedzmy mówię jakieś 40 minut i przerwa 15 minut. Powiedzmy wam, no właśnie, żeby to fizjologiczne podejście do tego siedzenia przy komputerze trochę ułatwić. Macie jakieś życzenia? Dobrze, no w każdym razie rozumiem, że wam wszystko jedno i tak będziecie zasypiali przy tych komputerach i tak. Tylko pytanie, teraz tak. Jeżeli ktoś na przykład zdecydowanie chciałby w trakcie wykładu coś mnie zapytać, no to łapka w górę, ja będę starała się patrzeć, prawda, że coś się dzieje, dlatego że w ten sposób jest to też sposób na taką przerwanie, bo to jest trochę monotonne. No, oczywiście ja mogłabym tutaj robić rozmaite rzeczy, żeby to było mniej monotonne, to znaczy nie wiem właściwie, co miałabym robić. No, mogę się wygłupiać, ale to chyba żeby no bo to jest ciężko, no ale okej, no w każdym razie przerwa na pewno jest wskazana i dla mnie, i dla was, przynajmniej jedna, a jakbym bardzo miała kłopoty z mówieniem albo włączeniem się, to będę robiła przerwę częściej. Zapraszam was do przerywania mi, zapraszam was do włączenia kamer tam, gdzie się nie wiem, jakbyś wszyscy włączyli kamery, to co by się stało? Zawiesiłby się ten system? Próbowaliście kiedyś? Dzisiaj w tej chwili mamy 43 uczestników. Jeżeli by wszyscy włączyli kamery, no to ja wszystkich nie zobaczę, prawda? Czekaj, to jest taka opcja, żeby zobaczyć wszystkich naraz, ale nawet nie pamiętam jak, wtedy przynajmniej ten system się zawiesza. Nie, właściwie kamera to nie, no dobrze się wszyscy naraz zaczynają mówić. Dobrze, okay, to I have to switch to English. Anybody remembers one thing from their last lecture? For example, what are the main differences between bacteria and viruses? Albo po polsku, co głównie różni bakterie i wirusy. Może jedna sama na ochotnika coś powie. Zamianie była taka...
No organella and no nucleus, and they can either have DNA or RNA.
Right.
So viruses also don't have any metabolism, and we cannot propagate them alone in the lab. They need living cells for propagation.
Right. And usually they are much smaller than bacteria, although there are of course a few species of bacteria which are also very small. But for many people, bacteria, well, whatever is zarazek, is more or less the same. So it is always important that viruses are completely different because they are not independent organisms. They are just like dead pieces of information until they find the right host, the right place to enter. Okay, so I will try. The last lecture was mostly to show you that there are many viruses, that they are very diverse, and I will come back to that last lecture, but I will start today with something which I perhaps should have talked last time. This is about structure of viruses, so I will try now to share my screen with you. Is it visible?
Yes, it is.
Okay, so I can make it full. So what we'll start today is the presentation about how the viral particles are constructed. And I was talking all the time about how diverse are viruses, and here I would try to show you that the diversity is based on some very simple principles. So, okay, this is mostly in Polish, but this is what it's about. I mentioned it already, that the virus is generally just a piece of genetic material or a program which is somehow wrapped, coated by a box, which is a protein code. Sometimes it's also something outside the protein code, and it is called envelope. And viruses can, apart from that, have their own tools, which can be also part of the virus particle. Some of these tools, some enzymes have to be taken into the viral particle to make that the propagation successful. So piece of genetic information, DNA or RNA, protein code, some more layers, some construction like you see in bacteriophages here are more complex. So I will start with just making clear what some certain terms means. Virion, it's a kompletna cząstka wirusowa, it's a complete viral particle. So all these things together, yes. Capsid is that direct protein code which is enclosing, covering, and protecting nucleic acid. And nucleic acid can be connected very, very tightly with a protein. This is sometimes made sort of nucleocapsid this way. And capsid is constructed from structural unit. And structural unit means just protein. And that protein, it may be it's the smallest functional part of the capsid, but usually the capsid is constructed from bigger pieces, let's say, which are constructed with individual protein units. So there is also such a term as a capsomer, and it mostly is about morphological unit of the capsid, which can be visible, for example, in electron microscopy. So this is a capsomer is constructed from structural units. So this is the basic elements, and many viruses apart from that contain envelope. And then the envelope is a lipid layer, and that lipid comes from the host cell membrane, but the virus adds its own material, usually quite few or many proteins, very often glycoproteins, and they are very crucial for the virus to be able to get, essential for the virus to get to the host cells. So that slide I have shown before. So again, that there are really quite diverse shape and size, and usually we divide viruses for those which contain DNA or RNA as genetic material, and they are enveloped on a non-envelope. So the simplest, the smallest also are usually those which are naked without any envelope, like parvoviruses. This is a single-strand DNA viruses, or some RNA viruses like picorna virus and calcivirus and so on. And from that we can move to big RNA viruses like coronaviruses, like influenza, or huge long filaments of filoviruses like Ebola. Okay, so that looks very diverse, but basically what is viral particle is, as I said, a piece of genetic material and a code. And for some viruses, that's it, the genetic material and the code, and this is naked virus. And enveloped virus contain more, the structure is more complex, lipid layer with protein, viral protein and glycoprotein, and there are some viruses which contain quite many proteins in between. So, but basically, what we can find, forms which we can find in nature are like that. So, all depends on the structure of the capsid. The capsid can be icosahedral, which is tościan in Polish. That means this is a sort of spherical form made from, I will show in a moment, looks more or less like that, or, the nucleic acid inside. And the second form, the other form, which capsid can be present, is a helical. And that both a form of capsid, that means there is sort of tube, helical tube, which is usually tightly connected with genetic material inside, and this is looks like that. And both of these forms of capsid can be just naked, and this is virus like that, or it can be covered with envelope and some other structural proteins. So naked, icosahedral, naked, helical, enveloped, icosahedral, and enveloped helical. These are practically 4 possibilities, although there are also some viruses which are a little bit beyond that classification. And okay, so again, naked icosahedral, so those small naked viruses like poliovirus, adenovirus, hepatitis A, virus A has that kind of structure. Naked helical This is rather typical for plant viruses, and the model for that kind of viruses is tobacco mosaic virus. Enveloped icosahedral, this is usually, most of them are big viruses like herpes virus, but there are also DNA viruses, but there are also there is also yellow fever virus and rubella virus. Yellow fever is a RNA virus. And enveloped helical, well, that's structure you should know very well because coronavirus is like that, but also rabies virus, influenza, or for example, virus oder measles virus. I mentioned complex. Pox virus has is very rich in terms of composition virus and contains more elements. Also, some bacteriophages are a sort of combination of structures. So about capsid. I will show for a moment Polish slide in Polish just to make it easy for Polish students and for myself to remember all of that. So they looked very different, that to Polish and English. I will show that a few times. They look very different, that helical and icosahedral capsid, but in fact they are very similar in some features. First of all, they are very symmetrical. They are organized in a geometric shape, and here it's a rod-like shape. It's very often also compared to, to klatki schodowej, like staircase, because it's not really a tube. It's a helicase, which is somehow climbing, let's say, and they have all some kind of rotational axis. So that helical symmetry is, well, that helical structure may be very long. And as I mentioned, it's typical for tobacco mosaic virus. I will tell you about the structure in a minute. Icosahedral is, well, it's a little more complex. It's sort of ball, spherical shape, but it's made from 20 faces, 20 sides, and we can find 20 vertices, 12 wierzchołków, or 30 edges, and there are those sides, those faces are made from triangles. It's actually I don't remember the name. Podejściościan foremne I trójkąty równoboczne. Anyhow, triangles, and well, what they also have in common, these capsids, they are made from so they are symmetrical, and they are made from identical proteins. So that it may happen that the whole structure is made just from one protein, but very often the more proteins components of the capsid, and they are this is all very compact structure, and it is in nature planted like that there's minimum genetic information to make a structure which have let's say maximum capacity to be to contain something inside, and something is of course nucleic acid. And it's also important that they have that self-assembly mechanism. That means that there are like pieces of puzzle. So if you and we of course use that in practice. So that means that they can be self-assembling in a, for example, in without really presence of nucleic acid parallel genetic material. If we express the components of capsid in other cells, like for example, in insect cells, these pieces very easily find each other and make that form which we call VLP, that means virus-like particles. For example, the vaccine against papilloma virus, which is used also as a vaccine against cervical cancer, is made in insect cells at the moment, some other cells as well, but insect cells, and just the genes expressing the main protein of papilloma is expressed inside the cells, and when those proteins are produced, they self-assemble in the form like that, and this is used as a vaccine. So all these units are somehow the same. They provide a sort of maximal contact between cells, and this is all can be very efficiently described in, let's say, mathematical, geometrical terms. In Polish. Let's move to that first form, and here the model is plant virus, tobacco mosaic virus. So as you see, this is just a single protein arranged with sort of constant relationship. It's certain amplitude and pitch that the distance from this particular disk. So such a tube is made out of protein and nucleic acid, and it can be presented like that, like a big helix, or like a number of disks stacked on each other, and each disk is constructed from single protein, single structural unit. If you cut something like that in half, you would obtain such a wall, and then your SSM can measure certain geometrical features of that structure. This structure has been, for TMV, has been really very, very well studied, and so that protein subunits can be sort of arranged around that sort of from a disk, a circle, and the disk are stacked, and that tube is created. As I said, it's not just a plain cinder tube, but helical structure. This is connected also with the structure of nucleic acid, which is also adopting very easy helical structure. The length of that varies, but that is in terms of TMV, the number of subunits per ton of the helix is about 16, and this is well, another examples of such a structure. So this is the whole tobacco mosaic virus helix capsid, but also animal viruses can have similar structure. So, for example, this is vesicular stomatitis virus, wirus pęcherzykowo zapalenia jamy ustnej, to jest wirus zwierzęcy, it's an animal virus, and you see that its capsid also has the same shape, the tubular shape, and the diameter and well, a lot of other let's say elements of the sizes and of this of the structure can be well measured. And here there is not only one protein, but there are two protein subunits which make the form. It's similar virus. This is similar vesicular stomatitis virus is a rhabdovirus. This is very similar to rabies, the virus Siekliźny, and here you can see how it looks at the electron microscope, that structure, and that internal nucleocapsid core is covered also by an envelope. So this is we have an envelope which contains G protein, and N protein is is forming the nucleocapsid together with more complex virus than TMV. Our famous coronavirus SARS-CoV-2 is also a virus which contains helical capsid, and that nucleoprotein is shown here as that kind of folded structure, and one of the main proteins, nucleoprotein N, is very tightly connected with RNA, and outside of that is an envelope with spike proteins and so on, and this is how it looks like in electron microscopy. Influenza virus also has helical capsid, but in this case the situation is more complex because the genome of influenza virus grippe is composed of several fragments, 8 fragments. So instead of one big tube, there are several smaller tubes, which helices, which are tightly packed inside the influenza virus particle. And this is just the models for a helical structure capsid. So this is again TMV, and the building, which is a little bit similar in structure. So sometimes the viral structures are reflected in the modern architectures. And now the second form of capsid, which is called icosahedral, which is Tościan. It's a very symmetrical structure, ball-like, let's say, spherical. And this is, if you analyze it from the, let's say, mechanistic and economic point of view, this is the best way of producing a shell of equivalent embodied helical structure. So we have 20 also, well, as I said, minimum free energy structure, but actually sometimes this this structure is composed of quite many proteins, so it is not always that simple. But generally, it's like that. We have structure which is made from 20 triangles. Equilateral. That's the word which I was missing before, and very symmetrical. So I can rotate it again around. Different symmetric, oh my God, oshysymmetry. I forgot again how to call it. But that structure can be also described very precisely in geometrical tests. There is also such, as you see, for example, in that case, that triangle is made from 3 different proteins. So actually, there is very often 60 proteins which makes such a structure. This is typical for picornaviruses, and you can see that 3 subunits are forming one triangle, and there is one other protein which is inside that structure. So there's the rotational axis can be somehow like here shown. That there are so many rotational axes which can be so we can divide that symmetry according to those axes. And this is more and more, and this is here I will not be explaining that, but the number of that smaller triangles or smaller or subunits which are part of the big triangle, and it's characterized by so-called triangulation number, which can be T1, T4, and so on. And the more complex, the higher is the number. But I think we can skip that, and have already seen that. And this is an example of against plant virus, tomato bushes plant virus, which is unenveloped icosahedral virus, and viral capsid is composed of 180 subunits of a single capsid protein. So not like in the virus which I showed before, when we have several proteins forming individual triangles. Here we have just one. Again, architectural form mimicking that viral particle. This is that was some world exposition in Montreal in 12th century. This is the United States pavilion, which looks a little bit like a virus. And this is how these structures look under the microscope. Here you have seen alpha virus. This is a plus RNA virus, and if you put it on a monolayer on a coated grid, put it on analyze it by electron microscopy, it looks like that. And here is the image obtained by cryomicroscopy. So this is perhaps a good moment to think how you can see how it can analyze virus structure. And well, this was the problem for a long time. They are so small, so people saw them growing even in cells, saw the effects of virus presence mostly in forms of disease or destroyed cells, but couldn't see the viruses themselves. Electron microscopy is the basic method which allows us to see the viral particle, but the analysis of the structure, the methods which is also very important is X-ray crystallography. So you can imagine that there is a dispersion of X-rays on the viral particle which can be crystallized, and then the picture can be reconstruction by image reconstruction. The modern method, one of the modern methods is cryo-electron microscopy. I'm not sure a few words about cryo-electron microscopy. You probably have heard about that method on some lectures, but well, the basic difference between that microscopy and the classical transmission electron microscopy is that it preserves native structure of a sample, because the sample is very quickly frozen in a very, very low temperature, as you see in well, much less than a second in a very low temperature. It's so-called vitreous A state, and that is requires special equipment. But that means our cells, our samples are based in water. So if the water is somehow preserved, that means that the natural forms are also preserved. And normally, for electron microscope, the samples are dried and treated with solvents like all kinds of chemicals and solvents, and then sometimes the structure may be a little bit distorted. So here it's a technically difficult method, but really brought us a lot of new information about virus structure. And this is traditional electron microscopy image of animal Biologia virus, like polio or virus pryszczycy or rhinovirus, the most common virus which gives us catarrh, runny nose. And the same structure shown as a single particle. And this is structures obtained by X-ray crystallography. But I will be talking about that structure on Biologia viruses lecture in a few weeks, 2 or 3 weeks. But and as you see, this is how it looks like in cryo-electron microscopy and image reconstruction. And that method, this is just what all these images show us, allow us to show different features of such a structure. So, for example, here highlighted antigenic sites. And I mean, that method allows us also to show what is on the surface of the particle. Here, all proteins are coded by different colors, and also can see individual protein here. This is organized. The image is shown like that by X-ray crystallography. You can see some of the interior of that particle. So all that image reconstruction, all this method combined allows us not only to evaluate the shape and the basic features of the viral particle, but also how it's organized in terms of antigenic sites and how it is also interacting with receptors. Here, this example of a capsid icosahedral from herpes simplex, Wirusów pryszczyci, which is also a capsid, but you can see this is very, very complex in comparison to those tomato Bushstan virus or poliovirus, because this is a big capsid, about 125 nanometers diameter, composed of 162 capsomers. So there is quite many proteins which take part in the formation of that capsid, and also the process of formation of the capsid requires additional proteins, and there is a scaffold, rusztowanie, and some individual proteins are collected on that scaffold. So here the multi-stage procedure to form such a capsid. And so, also this is again the herpes virus. So as you see, the structure is much more complex, because this is an enveloped virus with multiple proteins surrounding the capsid and multiple proteins in the envelope. And this is electron microscope, and here again as electron microscopy images showing us different, like looking inside that structure and on the and evaluating surface of it, and showing the distribution of particular proteins on the surface of the viral particle. Another complex virus is a retrovirus, HIV, human immunodeficiency virus. This is an artistic image of that virus with glycoprotein, which is called GP20, on its surface. This is a little bit like with coronavirus. They do not form spikes; they are more like round structures on the surface of the viral particle. And this is how it looks on the, let's say, more artistic image visualization. This is just a drawing, so you can see that lipid layer and the surface protein, and this is again that electron microscopy, cryo-electron microscopy result, analysis result. Okay, and just a few words about more complex symmetries. So, for example, pox virus, which are biggest viruses, they have a little more complex structure because they have outer and inner membrane. There is no typical capsid like that, but there are very independent viruses which contain very many enzymes. And also bacteriophages. You can see that bacteriophages are somehow like combination of icosahedron and helical structure and connected together. And so but generally, if you think about structure of viruses, the two typical structure, helical and icosahedral, presence of lack of envelope. These are the basic facts. Okay, so I think I will now stop at the moment, and we'll try to close this and open another, and then we can let me find it. And I will try to share this one. Okay, so after talking about the virus structure and the virus, it's on your screen, yes, viral proteins? Hello? Can somebody answer?
Yes, it is.
So I have seen, I showed that photo, that picture just a moment ago. Also, I have mentioned that some viruses contain much more proteins than on the structure of proteins. But okay, so viral proteins, what they do? They protect nucleic acid. Yes, here is nucleic acid, the most precious pieces of the viral particle, and it's protected by the coat, which is made of protein, capsid protein. And the other things which is essential for the virus is to attach to receptors on the cells, which can be permissive, can be the host for the virus. And here you can see that herpes virus, which is shown here, has as many as 14 different glycoproteins, which means that the virus can get into many kinds of cells. Apart from attaching, the virus has to penetrate cell membrane, and so this is also why it has many own proteins. But apart from that interaction with the surface of the cell, viruses need to replicate, and some viruses has to bring their own enzymes inside that particle. It's not the case for herpes viruses, but it's definitely the case for retrovirus. You remember that the HIV and other retroviruses, they need a reverse transcriptase. The virus has to transcribe RNA to DNA, which is reversed to normal form of transcription. And to do that, the virus has to contain, to bring that enzyme reverse transcriptase inside its own particle. Practically all minus RNA, which is opposite to mRNA, orientation of RNA opposite to mRNA, also has to bring their own enzyme. But viruses produce many other enzymes, and they also have many proteins which are focused on to how to take control on the cell host cells, and how to modify and weaken immune response. This is the table shows what viruses can do. So structural and proteins I already mentioned, the viral-associated enzymes, especially polymerases like reverse transcriptase or RNA-dependent RNA polymerase, and all many non-structural proteins, enzymes which are required for transcription, for proteases, for example. The same is for coronavirus. Some viral proteins are made first as a precursor forms, and the viral protease has to cut them into final form of the proteins. There are many regulatory proteins which are down-regulating expression of cellular genes, for example, expression of MHC class 1, just to make convince the cell not to show viral proteins on the surface. They sometimes contain oncogene, they sometimes contain all kinds of proteins which are important for tissue tropism, and sometimes they produce virokines. I mean, you know what is cytokine? All the molecules which are signaling molecules between cells, and the virokines sometimes are substances which may mimic cellular cytokines. So again, examples of such a viral enzymes, so I already mentioned enzymes like transcriptase, RNA, transcriptase, DNA-dependent RNA polymerase, and so on. But I will not be talking about it in details. Perhaps later during the later lecture. But just some things I would like to remember just now. That, for example, retroviruses have to have that RNA polymerase, like reverse transcriptase, which has also other enzymatic activity, has to be produced by the virus and brought by the virus itself. Also, hepatitis B produces such an enzyme, but generally, the bigger the virus, they have the more independent from the cell, and the more enzymes, the more proteins it produces. Okay, so let's move now to very simplified life cycle of some hypothetic virus. So we have a cell, and we have a viral particle, contains just DNA or RNA. This is here just one gene, but okay, this is very, very simplified. So there is virus, nucleic acid, a code, and that virus has to get into the cells. After entry, the genetic material has to be released and multiplied by transcription, and then obtain if this DNA that mRNA obtained by replicate. Also, that if this is DNA, because this is very simplified, this is just DNA. So that DNA has to be replicated and transcribed into mRNA. Translation, that mRNA allows for translation for production of code protein. Assembly of the protein together with newly replicated DNA brings us to new viral particle which have to exit from the cell. This is very, very simplified. And I like also that type of drawing, which shows that a little bit more animation. So first the virus has to find the cells, then to have to get inside the cell, then to has to release its genetic material, and using all the host function, what is necessary is to transcribe that genetic material and then translate it into proteins, and that proteins and the multiplied genetic material is necessary to obtain new viral particle, which usually requires some maturation, and they have to get out of the cells. Okay, and another drawing showing the same, introducing more and more details. So first entry is connected with sometimes with attachment on some structures which are on the surface of the cells, but and usually the enveloped viruses have more complex way of getting into cells because they may be also performing a fusion with the cell membrane, or they may enter the cell by endocytic pathway when the all kind of endosomes and the vesicles sort of swallow those viral particles, and the opening of the viral particle happens inside the endosome and not just on the surface. Okay, when after the attaching and penetration, the coat is removed. That is called uncoating. That getting out of that covering, and then we have next step, that is that which is connected with genetic material. And well, here it is not shown, but it may happen on a slide. It may happen all in the cytoplasm, especially for some viruses which are RNA viruses, which do not have to enter the nucleus at all. Other viruses have to enter the nucleus to obtain transcription of the DNA to RNA. Anyhow, viral genes, viral protein, assembly of new viruses, and water analysis. This is important because the exit may be connected with the with the killing the cells. When the virus escapes, the cell is already so changed and so completely devoid of its components that it's practically dead. And then the cell can produce even thousands of new viral particles. And some viruses do that in more restricted way, so that they do not kill these host cells, at least not from the beginning. And they are just, the viruses are the new viral particles sort of budding from the cells, and usually the number of viral particles is lower controlled. For example, of hepatitis B is only something like 40-50 viral particles per infected cells. So, just to summarize, what are the steps in the replicative cell of viruses? There is first attachment through a receptor, and well, here is the example for that CD4 is a receptor on T cells for HIV, and many other receptors for many other uses are known. Of course, okay, in a moment. And then the real entry that usually it is receptor-mediated endocytosis or fusion. Anyhow, any element, any step which allows to deliver the nucleic acid to the cell inside the cell, and then cutting. If it is the virus is in endosome, it is usually triggered by pH changes. You know that, for example, influenza and some other viruses, they require a low pH to open their code. And replication of viral production is the next step. First, some viruses have that sort of cascade of events. They're produced early proteins, the late production, and so on. And then usually structural proteins are produced at the last step, and then assembly and release. And just what I mentioned before, that some viruses, here is the example of Ebola virus, not a nice virus, but well, here it's just given like an example of a virus which is completely RNA, completely using only cytoplasm of infected cell. This RNA virus, the entry is by endocytosis, so the virus is taken by the endosome, and in low pH, the fusion between viral glycoprotein and the membrane of the endosome occurs, and then the RNA is coming into, is transferred to the cytoplasm, is transcribed and replicated. So everything happens inside cytoplasm. For DNA virus like herpes simplex virus, most of the, apart from fusion of plasma membrane, that the capsid comes to the is traveling through cytoplasm to the nucleus, and genome injected into nucleus, and transcription DNA replication is happening inside the nucleus, and then that's okay. And then there is many communication between. So somebody is trying to get into my room, and so the whole story is much more complex, because that virus has to acquire its structural proteins after the glycosylation in Golgi system, and so the whole exit is also quite multi-step. Okay, so this is comparison of two pathways in a replicative cell of the virus. Just a few words about that attachment. of course, what virus finds on the cell is not made for the virus. The virus exploits whatever is available, and this is very much connected with the tropism. I don't know if already we're talking about host range, but that means that, for example, some species, for example, let's say that it is a virus which can infect only pig, only mice. That is so the first fact that receptor is available. It depends on kind of the cells from this certain species, from the certain tissue. So there are viruses which only can infect only, for example, epithelial cells or respiratory cells or liver cells and so on. So there is that surface glycoprotein which are important for the virus, and there are also some integral membrane proteins which are involved in the entry of the virus. So the member of viruses uses some receptors and co-receptors, so that the entry process is multi-step. And this is just to give you examples of some such a structures, such a molecules which are exploited by viruses. So all kind of glycosaminoglycans and sugars, heparan sulfate, but there are also immunoglobulin-like proteins, integrin, which are normally also some integrins. Viruses like herpes viruses uses several different receptors. The same is hepatitis C. We already know what kind of that coronaviruses are also using the different receptors that are ACE2 and other. It depends on the kind of coronaviruses. But okay, that will be in the course of talking about individual virus particle. Just this is the simple fact that the virus can directly enter the cells, or it can be done by fusion with the plasma membrane, or fusion inside the endosome, and endosome can be also lysed, and then the virus travels to the nuclear membrane. So okay, that's enough from that part, and I will now I would like to make a break, and we after 10 minutes break, we will talk about what's happened after the virus already got into the cells, and how to propagate the viruses in the laboratory. Okay, so just for a moment. Thanks.
Yes, we are.
Okay. Well, the good thing about that sitting at home or sitting in my room is that we are not in our lecture room, the big aula, which is always so cold. I remember that people were bringing all kinds of blankets, but okay, and this is on the one advantage. Okay, so let's go back to that lecture. So now I'm going to talk a little more about what can we do with the virus in the lab. Let me share. Okay, you can see that, yes?
Yes.
Yes, fine. So I'm now going to talk about how what people invented to propagate the viruses in the laboratory, and of course the major dogma which I repeat many times, that viruses can replicate only inside living cells. So to replicate the viruses in the laboratory, we first have to grow living cells. And for a long time, it was really a problem, because really people tried to maintain somehow cells alive in the laboratory, and it didn't work. So for a long time, the only way really to make a passage of the virus was just animals, from mice to mice, from rabbit to rabbit. Well, but would be more difficult in case of human viruses which do not infect animals. And anyhow, growing viruses inside the animal just to multiply the virus. This is a really horrible procedure. So laboratory animals, of course, are used, and well, in our studies we try to really do that as well, minimize that animal use in the various studies, but there are sometimes necessary. I mean, this is, for example, if you want to try something which is connected with immune response, and that cell culture is not sufficient for that because you need multiple tissues, or you want to observe how the virus goes from one tissue to another, or you want to test a vaccine, for example. And this is very good if you have some animal model. I mean, not good for the mice, but mice are used in the laboratory all the time. It's mice, rats, guinea pigs. But there are still some viruses which do not grow in any other bodies and organisms than humans, apart from chimpanzees. This is the case for hepatitis C. And so the first trials on hepatitis C vaccine or drugs were used on chimpanzees, but practically it is almost impossible to do that in Europe. And in the United States, the whole big family of chimpanzees which have been helping with study of vaccines, they survived because the virus does not kill the animals. Okay, this is about animals, so we have to use them from time to time, but not for cultivation, for the certain line of experiments. The next way to grow virus in the lab are chicken embryos, or just simply chicken, that's just eggs. And in a minute, but the basic methods are all kinds of cell cultures, from primary culture via the so-called cell lines. So, and I will explain it, although you probably know more about the cell culture right now, and continuous cell lines. And first about those eggs. So I mentioned some the history of when I was talking about history of biology, about that situation after the First World War. That was when the people used embryonated eggs for prokaryotic viruses, and this is the method which is used until today, especially for influenza and yellow fever virus. And it's well, it's like that. Really, it's a real egg, and you have to put in the right position to find the to remember. Okay, so you can use different area of the embryonated egg. The chicken embryo about 10 of days old is the clue for that. So it's like one big cell, and the chicken embryo and all the membranes surrounded it are from the culture which we want to use for propagating the virus. So first, people make a hole in that shell, and then the viral material can be introduced different areas of that egg. Mostly, it is inoculation of błona chory antylantoinowa czy błona kosmówkowo-omoczniowa. This is the probably allantoic inoculation is the most popular. But sometimes amniotic ovulation, so maybe also yolk or directly to that sucker sounding the embryo. But anyhow, this is the place when so whatever is inside that egg, all the protein and so on, is the medium for the growing virus, and looks like that again. So so this is the procedure. First, you make a hole, then you inoculate with the viral material, and then after a while you attach a rubber to it and just collect the material with the virus. But also what is important, you can observe the changes. Here is changes made of that membrane, that chorioallantoid. Proszę. Jestem w trakcie wykładu. Dobrze, kończę o 12. So you can see that lesions or pox formed by pox virus on the that chorioallantoic membrane on the chicken embryo. That can be washed, that can be prepared, that can be fixed, and not only you can see that lesions, that pox, that characteristic changes in that membrane caused by the virus, but you can also count that. And you can also incubate it with, so you can see how the pox formation looks like, how that membrane fix looks like, but also you can incubate it with antibodies and check some presence of some antigens, for example. So here, this is just I think it's like that. So the membranes can be stained, and that can be stained and incubated, analyzed by immunofluorescent, for example, proteins. So to obtain vaccine, you really use million of eggs. So it looks like that, and collected the fluid from those eggs in the route I shown you. And this is because the influenza is very variable virus that has to be repeated every year. Although at the moment we have two kinds of influenza vaccine, one of which is grown in the traditional method with using embryonated eggs, or also in cell culture. This is Max Taylor, the inventor of yellow fever vaccine, and he managed to obtain live attenuated yellow fever virus by many passages of the virus in the chick embryo, and then finally has a live virus which could be replicated to some extent, but did not cause any disease symptoms. So this is called attenuated, and this is yellow fever vaccine. This is actually used. That particular strain is still used nowadays as a yellow fever vaccine. But generally, modern laboratories are based on cell culture, and cell culture was started. You probably all have heard about Henrietta Lacks. Her name is forever known as HeLa cell line. And really, people try to. Well, this is very interesting things what people do to maintain cells in the lab. Where the first candidates were lizards, because if you all kind of reptiles, because if you for example, that when a lizard, kiedy jaszczurka straci ogon, it grows back. So that people try to think, okay, that kind of cells can be cultivated because they grow in natural conditions. But okay, but that human cells were tried to be maintained for a long time, and somehow there was a miracle one day, because the cells from the biopsy from that unfortunate woman. There's a book about her, Nieśmiertelne życie Henriety Lacks, which is translated into Polish. I never could read that book from beginning to the end, because what was being done in the hospital in that time was really sort of torturing people, all that therapy, radiotherapy, and so on. Anyhow, that lady died, but her cells were unique. They reproduced at a very high rate, and they are in our laboratories until now. But generally, the cell cultures are generated like that. Some cells are taken either from adult animal organism, or from the embryo, or from the egg. That has to be mechanically dissected, chopped, and then digested enzymatically. And if they are only not completely digested, they can be used for organ culture, or can be used for cell culture or primary explants. So explants are just bigger piece of tissue, and this is something which doesn't long for a long time, but enzyme digestion allows us to isolate and select individual cells, and that she was treated with enzyme to separate the cell, like trypsin, and then cells are suspended in the culture medium and put in the vessel when they can attach. Normally, primary cells grow on monolayer on the glass or plastic container, and only cancer-transformed cells can grow on top of each other and do not form a monolayer. Monolayer, hodowla jednowarstwowa, is a basic form of growing cells in the lab. In virology, we sometimes have to use primary cells, which are explanted directly from donor organism, like for the human skin, for example, and their life is limited. They give the right conditions to study certain procedures, but they do not continue, and this kind of culture is not very reproducible. Sometimes this is only primary cells still maintain certain characteristic. Here is cell that is beating cilia, or some lymphoid cells which normally do not they undergo only limited number of divisions before they before they are too old, and so on. But okay, this is very specific. For example, primary cells are used also now for coronavirus SARS-CoV-2 to study what's happening in our respiratory tract and lungs. In our laboratories, in many laboratories, our favorites are so-called semi-continuous cell lines. Secondary cells, we call them cell strains. They are something in between primary and continuous cell lines. So there is a certain group of cells which are somehow selected from the primary cells, which grow for a longer time in culture condition. They divide, grow for 30, 40, up to 100 generation, but after a certain time they will started to change, and they get older and die. But during that first, let's say, 50 generation, they are much more similar to natural cells than for example, transformed cells. And for example, you used here cells isolated from bovine trachea to grow bovine herpes virus, and many other cells. This goes like that we have a frozen stock of these cells, and they make passages for let's say 40 times, and when they seem to be already tired, let's say, then we go back to that first stock, which was frozen in liquid nitrogen, and use them again. But the most common cells are cell lines, immortalized cells, which may be cancer, tumor cells, but they may be transformed in other ways. I already mentioned that the HeLa cells are the examples of such immortalized cell lines. They are always adherent, that means they stick to surface. They have also certain they spread across the culture, and then when they have these cells, HeLa cells, even if they are generated from the tumor, they have that feature which is called contact inhibition. That means the signals when the two cells are very close to each other, they touch, they stop growing, they stop divide, and this is important, because then we have to transfer them to a new vessel. Okay. These are example. This table shows what is important, what are advantages, disadvantages of different cell lines. So all that cell lines, which cell cultures with finite time life, with finite timing, like primary or semi-continuous cell lines, they have normal numbers of chromosomes, so diploid. While continuous cell line transforms have very often change a number of chromosomes, and they may be also tumorigenic. So certain cell lines cannot be used, for example, for growing vaccines, because maybe contain some, but they never used for product vaccines. But for some studies they can be used. They differ in that anchorage dependence, w zależności od przyczepienia się. Those more natural cells, they really need to be attached to the surface, while continuous cell line may be also growing in suspension. So this is on a monolayer, this is on, they may be also grown in suspension. HeLa cells can be grown like bacteria in big flasks. And here, these more natural cells have very high requirement in terms of media. They need a lot of serum in the medium, and they grow rather slowly. Well, generation number is limited. Here is the opposite. Their requirements about the serum are low, but they grow rapidly. Usually, the doubling time is about 12 or 24 hours. That means we have high yield. We want to have a lot of virus. We need a lot of cells. So high yield, for example, 10 to 6 cells per meal is a really a good meal, and this is why we use continuous cell line a lot. So cell strain will stop growing after some time, months, for example, and cell line will be growing and dividing indefinitely. So if you start cell culture, we start with a single cell, then it makes a colony, and those colonies are plated. And then if you look, you have cell culture laboratories already, so you know a lot of the cells. I'm just giving you as biologist's point of view, because cells are our tools. So usually, if you think about cells, the morphology, well, yeah, they're usually fibroblast-like or epithelial-like, and that doesn't mean they are really fibrous or epithelial cells, but they look like that. So this is, for example, baby hamster kidney fibroblast, but this is the culture when there are not very many. They seem to be still rather separated. But if you see the same cells in later, when the cells are confluent, they completely cover the whole surface of the cells. Here, the cells are epithelial-like morphology, and then this is the culture when they are still not growing very densely. Here is the cells when they do not divide anymore, and here astrocytes, so nervous cells, from the nervous tissue cells, and so this is some kind of mouse fibroblast. So you can see that the morphology of the cells look like that, and we very often talking about epithelial of fibroblasts, but they are not always. They look like that. You probably are familiar with the procedure how to grow the cells. So if they grow in the monolayer, in that adherent monolayer, from time to time, there is already situations like that, the whole surface is covered, and you have to make the passage. That means you treat them with trypsin. Cells can detach, and then you put them into fresh medium, and then put on a fresh bottle or well or plate, and then use them again. So these are the famous HeLa cells when their nucleus are stained with Hoex, blue dye. And this is just very shortly to make you familiar with some names, cell lines. So very similar. I already mentioned baby hamster kidney, but HeLa cells, all kind of mouse cells, vero cells that are very often used. If you look at the morphology, you can see fibroblast, sometimes lymphocytic, but mostly it's epithelial or morphology, and origin is, for example, here they from human lung or and this is embryonal from embryonal tissue, and there are also many animal cell lines, for example, from the one which is very widely used, for example, for production of monoclonal antibodies, which are used in many therapies. This is so-called, sometimes say CHO cells, Chinese hamster ovary, they are very widely used in many laboratories as a for production of different proteins, but mostly animal, mostly monoclonal antibodies. So you can see the characterization of cells is the morphology, the origin, the age of the samples are either of embryonal tissue, and was the tissue normal or neoantigen. Sorry. And also characteristic, it is susceptible to viral infection of not. It is rapidly growing, can be grown in suspension. So this is the most interesting as in cell line. Well, all these cells have certain requirements. This is what we need, essential amino acids. We have this, what we need to live, survive, and grow. The same is needed for our cells in the culture. So we have to supply the cells with all the essential amino acids. We have to supply them also with vitamins and glucose salts and so on. But for mammalian cells, what is really important is the serum. And that serum, which is 5 to 10% of total volume of the medium, it has to be bought. And okay, for bacteria, we can to some extent make can imagine, because we buy all kinds of media, but we can collect all the components of the media, make that medium in the lab. For cells, that would be much more difficult, because the serum, which is in that essential component and most expensive component of the medium, contains all the things which are very difficult to provide in synthetic way. So hormones, lipids, proteins like albumin, fibronectin, growth factors, many other metabolites which are in that serum, that's essential for cell survival. So we have to grow cells in the laboratory. We need special laboratory. We need media. What is here? What I well, just also to remind you, you know the names of the media, but this essential medium, it's the component. It's formulated for the HeLa cells and lung cells, and that the next generation from Dalbecco. Dalbecco is a well-known name of genetist and virologist, and they contain, for example, 4 times more vitamins and more glucose and so on. There's DMM with high glucose. RPMI. This is also very commonly used medium. Okay, I can skip that. What's happened? Okay. Now this is just to show you that this is really important. The media for cells are very complex, and well, they're expensive, and this is components. And some of the names which are very commonly used in virology, I mentioned HeLa, I mentioned already, CHO, well, it's not the right way to say, BHK, baby hamster, also Chinese hamster ovary, many mice, murine cells. These are cells which we use very often in virology, but we also use some very specific, for example, for growing hepatitis viruses. We use all kinds of HUH, that means cells, HUH cells, which are derived from liver hepatoma, liver carcinoma hepatoma. Again, so you can see, for example, HeLa is very commonly used for many viruses, but for poliovirus, adenovirus, and so on. We use very often MDCK. These are Marpin Darby, that is her name, canine kidney. We also use MDBK, Martin Darby bovine kidney. These cells are used for influenza, growing for influenza. And I mentioned Vero because Vero are also very often used for growing all kinds of reagents and therapeutics. So they are also very flexible. So many viruses like measles, mumps, rotavirus, but also herpes virus can grow in vivo. Okay. And where you are familiar with that kind of vessels, if you had classes in cell biology, so we use all kinds of bottles. We call them usually T bottles, and all kinds of plates and multi-well plates. And multi-well plates, the very common are 6-well plates, but there are also those which contain 96 or 12 or 24. And for those cells which can grow in suspension in liquid culture, that we use that kind of vessels which have some sort of mixer, put a magnetic mixer, and they have that advantage that you can add some components to that culture and remove without stopping the culturing cells. Sometimes we really need a lot of cells, and to grow a lot of cells, people invented all kinds of small devices to increase the surface of the vessel, because if the cells, I know this is very difficult to show, because normally I would show you things from the laboratory, but perhaps it would be better to include the movie. I could do that. I didn't think about it. Just not okay. So still with pictures. So if you, for example, have a round bottle, you can put that kind of extension inside the bottle, and then using minimal amount of medium, that bottle will be rotating. This is the apparatus when you rotate that kind of bottle, and cells will be growing not only on the normal surface, but on that additional surface formed by the structures. So there is, and the amount of medium, expensive medium, is not that different. So this is well, a kind of devices, from that simple bottle to all kinds of fermenters. And the other method which was used, I never used it myself, is used microcarriers. So there's a suspension for that kind of plastic microcarriers, and they are like each one is covered by cells which can grow on the plastic round structures. You can see that cell growing on such a microcarrier. Another reason to increase the number of cells. Sometimes we need very specialized culture. For example, polarized cells. Some viruses can enter the cells only from apical medium. For example, if you can imagine, these are gut cells. I mean, those epithelial cells which are lining our intestines, and they have that characteristic organization. So they have two different surface, that apical membrane and basolateral membrane, and virus, some viruses can enter the cell only from that surface, and then they can very often spread from cell to cell by that tight junction or other connection between cells. So if we want to preserve that natural organization of that kind of cells, we need to use porous filters and two kinds of media, which are this is just multilayer canine kidney cells. Of course, they will be growing on normal condition as well, but if we want to take advantage of this polarization, we need to use something like that. You see, this is the dividing and this case look like that, and if we create real conditions to polarization, then we obtain that kind of polarized monolayer. Just to remind you how to preserve cells in the lab, they are preserved in liquid nitrogen -80 degrees. Usually the cells are trypsinized, then kept in glass or plastic vials with some preservative like DMSO or glycerol, and they are very slowly cooled from first in the refrigerator, then -70, then they are sealed in the glasses and kept in liquid nitrogen. We have to keep very careful records, and then if we want to need the cells, we throw them rapidly and put them in the culture, back in the culture. Well, there's our enemies, all that beautiful, rich medium containing all kinds of goods, sugars, amino acids, growth factors. Other organisms like that too. So bacteria, of course, mycoplasma, fungi. They love to grow in the same conditions. So we have to be very careful. And why we have to work in very sterile conditions. Sterile means you have to use laminar flow like that. This is a chamber which can be either open from the front. This is when the want to protect just our sample. But if you also want something like that, so the air which comes to the chambers is filtered, and this is the direction of the air flow. So the preparation is safe, but the person who is doing the experiment is not that safe. So this is why the biosafety cabinets which we work which we use when working with us are different. We have always that glass in front of you, and the air is filtered twice by HEPA filters, which capture all viruses or small particles, and that airflow goes like that and is filtered constantly. So the person who sits in the front of that in the chamber is separated from its contents by that glass. This is typical multi-well plate. And if you want to work with dangerous pathogens, you need increased biosafety level, BSL. And at the moment here on our third floor, that BSL level, which was something like between 2 and 3, one lab is adapted to something which will be between 3 and 4. The safety level is the highest. And if you work with the virus like Ebola, then this is the kind of laboratory is necessary. That means that the laboratory is completely sealed, and the pressure inside is lower than the pressure outside. So if there is any breaking in the wall, the air will be sucked inside, not put outside. All fluids, all leftovers from that lab has to be sterilized. And this will also, actually, this is what we do in our laboratory right now. I mean, all the materials used in the lab are sterilized, but the laboratory which is constructed right now will be completely sealed, and that means that people have to also work with the probably again with that kind of uniforms. That means everybody has an individual filtration system. Not like a diver. It's not as in a container with air. This is just filtration of the air, so you have beautiful clean air to grow. So this is that kind of facilities which are at present required for working with SARS-CoV-2, and this is why we are remodeling one of our laboratories. Although after discussion a few weeks ago in Italy with people who have all kinds of laboratories like that, at the moment the laboratory still has to be sealed completely. So that means that there is no way. There are special door and there are special steel walls. But for the experimental, probably some restriction will be a little bit less, because we are all vaccinated now, and using just normal medical mask would be enough. But at the moment we plan to use it like that, and that laboratory will be completely close to people from outside who do not have training how to use it. So just coming to the end of that lecture, and this is yes, do I have some time? Okay, so the next thing is what is happening after infection of the cell, and well, I already mentioned that after and how we analyze, how can we detect the virus, how we can characterize the virus, and we are also talking about diagnosis. So it all depends on what kind of virus, what kind of cell, but there are basically two different approaches. When the cell is damaged by the virus, we call it cytopathic effect, or not damaged, we can use different ways of detecting the virus. But generally, what is happening, do I have that another way? What can happen after the virus infects the cell? First of all, what may happen is that light lytic infection. That means that virus infects the cells, produces new viral particles, progeny, and way doing that is killing the cells. and that cycle takes, well, sometimes 12 hours, sometimes longer. But many reasons why that happens. But sometimes there is persistent infection. That means that virus is produced, but the cell is not killed. Cell may produce virus for months while still multiplying. Latent infection is completely different. That persistent or chronic. So this is chronic infection, typical, for example, for hepatitis viruses. Latent infection, that means that virus is hidden, and so after primary infection, the virus is traveling to specific places in the body, like in case of herpes simplex, to neurons, and they become latent. That means they do not produce infectious particles. They just the genetic material is inactive, but that latency can be broken by external factors, and that they will be talking about that in details. And the other result, so this is latent infection, persistent or chronic infection, latent infection, and in case of some viruses, it can be transforming. That means viruses like some papoviridae, adenoviridae, some herpes viridae, retroviridae can transform the normal cells into well, sort of cancer cell. Transformed cells are capable of unlimited cell division. This is the first stage of cancer cell, and this is in some cases it is also accompanied by integration in retroviruses of viral genome into the host cell. That's only in and there is also so-called abortive infection. That means virus can induce effectiveness. That means that the virus gets into the cells, but there is something lacking in the cells, and maybe although the part of the replication occurs, anyhow there is no virus progeny, and that is means that even if the virus is present in the cell, there is no spreading infection. So this is again to some perhaps some words first about latent infection and cytopathic cytopathic effect. That means that virus is destroying the infected cells, and how that virus is doing, it depends. So this is Polish. Okay, I will try to translate it very slowly. I'm sorry, I still have some slides which I've forgotten to translate. How the virus, why it's happened that virus kills the cells, why it destroys. Well, there are some, I will show you later on piconebulosis how quickly it happens. Sometimes that already 2 hours after infection, the cell is completely under control of the virus. All synthesis of cellular proteins is stopped. We call it shut down or shut off. Virus that the cell, instead of producing its own cellular protein, producing only viral proteins. It may happen because virus is degrading cellular mRNA, or it is just producing so much of its own RNA that it is somehow prevailing the cellular translation. The other things virus can do is to stop synthesis of cellular DNA, and this is, for example, big viruses like pox viruses can do that. They also can stop splicing and transporting of RNA from the nucleus to cytoplasm. Apart from that, the viral proteins are accumulated in the cells, and this is a very toxic effect of the cells, especially if you, for example, think about many glycoproteins, which are components of viral envelope. They accumulate in the all membrane inside the cells, and that, of course, it's modifying, destroying their activity. So the all cell membranes are well becoming more. Well, they have holes. They have a completely different mechanism of ion transport. And well, also the other thing, how the virus is destroying the cell is not direct. Very often our cells are destroyed not by the virus, but by our immunological system, because when our immunological system notices foreign cells, viruses on the surface, then this is the target of attack of antibodies and T cells, which leads to the destroyment the destroyment of the cells. Okay, that's also the cell is destroyed by the changes of destroyment of its cytoskeleton. The infected cell become unable to attach to other cells and to the surface of the vessels. All this of that can be observed in the microscope. Many of these effects, like change of the shape, like the fact that the cell is not longer attached to the surface of the vessels, and this is all a cytopathic effect. Final cytopathic effect is the killing of the cell. And this is just to show how. So this is an infected secondary culture of hamster embryo cells, and this is like confluent cells because they grow very, very close to each other. And after infection with a polyomyvirus, you can see that there is great change and that cytopathic effect is obvious. Here is the same. The cells, there are Vero cells infected with herpes simplex. The cells became round, and first you can see the first slide of the signs of plaque formation. Here will be a plaque, and here you can see that there are empty areas in the monolayer. Well, this is show how it might happen gradually. So uninfected cells, then infected with a small amount of virus, so-called low MOI. I'll explain that in a moment. And then so you can see single plaques. But if you put a lot of virus into that culture, all cells become infected, all cells become round, all cells are floating in the medium, and you do not see the normal culture anymore. And that's perhaps time to explain some terms, and I will finish with that. Cytopathic effect that I already mentioned. We will be talking also about virus titer, Mianovirusa, about something which is called TCID50, that's tissue culture infectious dose, plaque and plaque assay, multiplicity of infection, AOI, plaque forming units, PFU, permissive cells, and I probably will be mentioning very often something which is called ELISA, enzyme-linked immunosorbent assay, and I will try to explain all of that next lecture because I think it's already 12 o'clock. Yes. Okay, I know this is more technical and more practical, and it would be much easier if you just could see some of these things in the laboratory, and some of you will see, because some of you will start very soon. I don't know, perhaps it's only in winter virology class when we'll try to show you how the virus grow inside the cells and how the cells look when they are infected, just basic facts. And of course, you can see a lot of that in the internet, and perhaps I will try to show you some movies. But I don't know about you. For me, this with practical things, it's quite different when you touch it with your own hands. Oh, sorry, where is that one? And so this is a well, sort of dry way of introducing you into virology, practical virology, but I just wanted to give you some information on what we are doing here and how virologists work in all the laboratories, and why also this is quite difficult science, because you need to know basic molecular biology, microbiology, cell biology, and you need a lot of money to work with viruses, because all that sterile equipment, sterile plasticware, media, and many other materials are really expensive. So this is a limitation of a virologist's life. Thank you very much, and when we are going to meet next time, I don't know. Yes, it's already in November 4. So I will see you in a week, and then we'll be talking a little more still about the practical things and about how to detect and recognize the virus, identify the virus. Any question for today? Okay, so thank you and see you next week.
Thank you. Bye.
Thank you. Goodbye.
Thank you. Goodbye.